Good afternoon, everybody. My name is Jasmine Zhou, and I'm a professor at UCLA. Today, I'm going to introduce EarlyDx, a company spin-off from my lab. And we consider us as a preeminent leader in liquid biopsy. And our first product is detect and locating cancer early. So as we know, the best way to fight cancer is to detect it early. However, there's not a very good method in detecting cancer early. So there's several, screening products for a couple of cancer, but the compliance rate is actually very low. So to detect cancer early, we need a product that is, very convenient, like a blood drawn, a tube of blood. And it should have high accuracy, high sensitivity, high specificity. And also that it should cover the majority of cancer. And in addition, if it's affordable, cheap, that'd be great. And the market is huge. It's global market more than $100 billion per year. And it's still growing. Now, our product is called MethylScan. That meets all of these criteria. And we think our best-in-class liquid biopsy test at the moment. So how it works? So we draw the patient, from patient we draw a tube of blood. And from blood, we extract cell-free DNA. Those are DNAs that are floating around in the blood, and that is actually, released by the dead cells, including tumor cells. And we sequence those DNAs, and from which we detect the cancer DNA. Because cancer is actually genetic and epigenetic disease. The difference is that cancer DNA and normal cells DNA that determines a cancer cell to be a cancer cell. So we need to basically find the needle's high stick to find the tumor DNA out of the cell-free DNA in the tube of blood. And we invented technology called MethylScan that can sequence the cell-free DNA methylation genome-widely in a very cost-effective manner. And from which we build the artificial intelligence platform to use all the genome-wide methylation data and to detect cancer and locate which organ actually has cancer. And our method has been published in top journals, Nature Communication, and our clinical study, we have tested on around 2,500 patients. And we have excellent results to detect not only the all cancer stages, but also early stage cancer. So compare us with the leader company Grail in the space. You can see in detect the stage 1 cancer at the specific very high specific 99.3%. Our data showed that we can detect stage 1 breast cancer at a sensitivity about 50%, while Grail only have 6%. And for all the major cancer type I list here, there's a big margin will lead in Grail's product. Also in terms of function, because we actually get a genome-wide methylation. Genome-wide methylation that's basically include biomarkers, methylation markers of almost every disease. So our product can not only be used to detect cancer, but also detect many other diseases. And that's basically the data that's become a big data problem. If we have a lot of data, if we can push this product into cancer detection, then we'll get a lot of data from a big population, and from which we can train the classifier to detect cardiovascular disease, autoimmune disease, and many other diseases. And also, the material cost is very low. Our material cost per assay is only about $50. And Grail sell its product right now $950 per assay. And also we need very little blood. And here is showing that we actually secured prospective cohorts for testing our product. For example, we got access to National Lung Cancer Screen Trial. So that's the largest and the most, famous lung cancer screen trial. And we got access to the biobank, me and my UC5. Okay. And several other cohorts. Okay. So we can commercialize by partnering with other CLIA labs. And that's all the grants we got from NIH. Every grant is peer reviewed. Okay, that's fine. And what I'm trying to say is that
we should probably leave some time for the questions. Thank you.
Okay.
I think one question I will ask, is there something really shortly that you want to cover before we ask questions? Anything you want to say?
Oh yeah, I want to say that you can see that we can actually we are working on other diseases as well, not only cancer, but also NASH, some disease and transplant monitoring, for which we all secured collaborators already. And that's the leadership. I'm a professor at UCLA, and my co-CEO.
Okay, this we
Greg.
can see already. Can you say what's the patent on what is
we
the
have
technology?
a patent portfolio, actually all managed by Wilson Sonsini. We have 7 patents to have granted.
And what is it covering? What's the patent on? Is it
one
on
cover the
algorithm?
computational, one cover experimental technology.
And the sequencing, are you using generic
Oh,
sequencing
no, we have
devices?
our own library preparation. We use Illumina, we can use any sequencing machine, but the library prep is patented.
Okay, and then can this technology be used in any CLIA lab, or these samples have to be analyzed at your...
any CLIA lab, but we actually have established our own CLIA lab in Agua Hill, Calabasas,
Well, it's quite amazing. I think we've all kind of lived through Theranos and their claims there. So I'm just trying to understand how you were able to achieve what you can with that low of volume and at that low of cost.
Because we developed, it's unlike Theranos, we have a scientific, high-profile papers to, introduce our library preparation so that we enrich only self-reading fragments that carries methylation information. We don't sequence every self-reading fragments, but only those that carries methylation information. And that dramatically lower down the cost. We only have the cost of 1% of what other people would do for whole genome methylation sequencing. And because we got a methylome information, we can aggregate all this information. So we don't need a lot of blood. We don't need a lot of genomes in the blood. So we only need a low volume of cfDNA, but we get the most information out of it. Most relevant information out of it.
The specificity and the sensitivity test that you cited earlier, this is on the general population, or who's the sample that you got this kind of data on?
Okay, those are the case control here. Case control, that is normal patients that carry a lot of this disease. For example, when we change this classify, that is the blood collected from UCLA, from liver clinic. These people have a liver cirrhosis or liver cancer, and we also got the lung nodule people. People has a lung nodule, benign or malignant, and we also source the commercial vendors, the general population and the cancer cases.
So let's take the pulmonary nodule case. Then a lot of people get a CT scan. There's a pulmonary nodule. So that's one of the subsets you have.
Yes, that's going to
So
form
you run your analysis, and if it turns out it's positive, specificity is pretty good. But if it's negative with a sensitivity of less than 50%, now what do I do with that?
Okay, so if it's then that means that sensitivity. So I think in this case we could at least when we compare GRAIL, we have to, this is screening. So we have to get a very high specificity so that we do not load the medical system. However, for lung nodule case, in that case, we require high sensitivity instead of high specificity. Because in this case, there's already people with lung nodule. So there's not a lot of patients base that, but the patient want to know, if I don't have to go to biopsy, I want to make sure that I actually really don't have cancer. So that's different cutoff.
Using this particular screening, what is the sensitivity level that you could change if you decide to increase the sensitivity rather than maximize specificity?
Yes,
like that?
We have actually a slide I don't have here. It's already over time, but I have a lung nodule specifically. And,
but just give me an estimate in terms of, is it 90%, 80%, 50%?
Oh, for different cancer, it's different. For example, for breast cancer, it's, depends on, if you want to require very, like a CT scan, this high sensitivity, we still have about 10% margin than its specificity. So it's in terms of lung. So we actually have a lead product, is a lung nodule determination and liver cancer cirrhosis HCC screening. So we have these 2 lead products.
So in practical use, would you see this as being something like when you go for your annual visit and you do a blood test, then you would do this screening and determine? Yes, very affordable and annual screening. And so right now we don't have a lot of longitudinal data, but when we incorporate longitudinal data, you will get even more sensitivity specificity.
Let me follow up on that question though. So you go in for screening and you're not one of the cohorts. Now your sensitivity and specificity are not defined for that population. So what do we do with that data?
So excuse me.
You said specifically the sensitivity and specificity are for those people that you identified.
Yes.
The question earlier was for the general population that comes in and wants a screening, you're not going to get those kind of numbers. So what is the use for the general population?
Oh, okay. So, I mean, those are the case control, right? And, but I think, it has to represent a general population. So those, that it has some high risk patients, but also has, healthy people. And in general, we cut off the 50-year-old, all the people here are above 50-year-old. And so we think it's represent general population, but because we have high risk, we actually harder. It's a bit more harder, problems than general population here. So of course, finally, we have to do prospective cohorts. And that's what we actually working with VA right now to establish prospective cohorts.
Yeah, potential use cases if you have a very high risk group, be it somebody with cirrhosis or be it somebody that has multiple nodules or whatever it might be, you could use it to see if you could potentially detect a early onset.
Yes.
Right, because your specificity is high.
Yes.
But it's a screening to detect whether or not it would not be that effective.
Yeah. So in that case, like for high risk population, we don't need that specificity. We need a high sensitivity. So that's a different scenario. I'm talking, if I compare with GRAIL, that's a screening population. So we have to have a high specificity. Less doesn't matter. I mean, sensitivity is less crucial. Actually, it's crucial, but for insurance to approve, we cannot have this. Yeah.
How far away you are from going to market?
Very soon, because our CLIA lab is undergoing certification right now. So we're looking for, we actually, so we already secured 25 million from NIH. And we, for developing and translating the product, we're going for another 9 million for phase 2B. And we have raised so far $10 million from VC. We're raising right now 7 million. We already secured 3.5. So we actually have another 3.5 volume for this round, closing June 30th. And be happy to discuss.
Congrats on that. Thank you very much.
Thank you.